Good morning, everyone, and welcome to today's presentation. I'm Dr. S.K. Singh, and I hold a PhD in this field. Today, we will be discussing the qualification of dry heat sterilizers. I'm excited to take you through this important topic. Over the course of this presentation, we'll cover the key aspects of sterilizer qualification, including the processes and standards involved. So, let's get started and explore this essential subject together. Let's explore the basics of dry heat sterilization. So, what exactly is a dry heat sterilizer? Well, it's equipment like a hot air oven or a tunnel that uses hot air through convection or conduction to sterilize or depyrogenate items. Think of items such as glassware or metal instruments, those that can withstand high temperatures. It's important to note that validation is a crucial step here. We need to ensure that the sterilization process is consistently effective every time. Now, as for applications, dry heat sterilization is commonly used for depyrogenation of glass vials, sterilization of stainless steel parts, and for materials that are heat stable. This method plays a vital role in ensuring the safety and sterility of critical components in various industries, aligning perfectly with our overall focus on sterilization techniques. Let's talk about the regulatory expectations surrounding sterilization equipment. Regulatory frameworks like the FDA, the EU GMP Annex 15, and ISO standards, such as ISO, for dry heat, all emphasize the importance of properly documenting the qualification process. This documentation is crucial because it provides evidence that the equipment meets the required standards and is fit for purpose. When we perform IQ, OQ, and PQ, we're covering three key phases: installation qualification, operational qualification, and performance qualification. IQ ensures that the equipment is installed correctly and calibrated properly right from the start. OQ then verifies that the equipment operates as intended under specific, defined conditions. Finally, PQ confirms that the equipment consistently performs its sterilization functions, whether it's biological or endotoxin removal. The main goal here is to ensure that the sterilization process is reliable and effective. We want to be confident that the equipment is installed and functioning correctly, that it behaves predictably during operation, and that it consistently achieves the desired sterilization outcomes. All of this ties back to our overall theme of maintaining quality and compliance in sterilization processes. Now, let's take a closer look at the qualification lifecycle. This process is typically divided into three distinct phases: IQ, OQ, and PQ. First, we have the IQ, or installation qualification. This phase ensures that all equipment and systems are installed correctly according to specifications. It's about verifying that everything is in the right place and functioning as intended from the start. Next comes the OQ, or operational qualification. During this stage, we test the equipment under normal operating conditions to confirm it performs consistently and reliably. It's about validating that the process controls are working effectively and that the system operates as expected. Finally, there's the PQ, or performance qualification. This phase assesses whether the system can consistently produce a product that meets quality standards over time. It's the ultimate test of whether the process is truly validated in real-world conditions. What's really important here is that these phases are executed sequentially. Each step builds upon the previous one, creating a robust trail of documentation. This documentation is critical because it builds regulatory confidence in our process controls. Essentially, by following this structured approach, we ensure that our processes are not only compliant but also reliable and repeatable, which is vital for quality and regulatory approval. This lifecycle approach is fundamental to maintaining control and assurance throughout the product's lifecycle. Now, let's move on to installation qualification, or IQ for short. The main goal here is to demonstrate that the dry heat sterilizer has been installed correctly, in accordance with the original design and the user requirement specification, which we often refer to as the URS. Essentially, we want to verify that everything is in its proper place and configured properly before we move forward. This step is crucial because proper installation lays the foundation for everything that follows, including validation and ongoing operation. During IQ, we'll check that all components are installed according to the specifications and that the equipment matches what was initially approved. This includes confirming that the correct parts are used, that the installation environment is suitable, and that the equipment is set up to meet safety and quality standards. By thoroughly documenting this process, we ensure traceability and confidence that the sterilizer is ready for the next phases. Remember, a solid IQ helps prevent issues down the line and supports the overall integrity of our sterilization process. So, in essence, IQ is about giving ourselves peace of mind that the equipment is installed properly and ready to perform as intended. Now, let's move on to operational qualification, or OQ, which is crucial for confirming that the DHS operates consistently and stays within its design parameters. The key activities here include temperature mapping of the chamber using calibrated thermocouples. This helps us ensure that the temperature distribution is uniform and meets the specified conditions. We also verify the alarm systems, particularly the high and low temperature alarms, to make sure they function correctly in case of any deviations. Additionally, we check the airflow and circulation patterns within the chamber to confirm that heat is evenly distributed and there are no cold or hot spots. Regarding temperature mapping specifically, we spread sensors across the chamber, anywhere from 16 to 25 points depending on its size, to get a comprehensive picture. During the cycle, we log the temperatures at these points to detect any inconsistencies or areas that might be cooler or warmer than ideal. This thorough process helps us validate that the chamber maintains the proper conditions consistently, which ties back to our overall goal of ensuring reliable and safe operations. Now let's take a look at an example of acceptance criteria. In this case, we're checking that all sensor readings fall within a specific tolerance range. For instance, during depyrogenation, we might specify a temperature tolerance of plus or minus 5 degrees Celsius around 250 degrees. This helps ensure that the process is consistent and effective. Next, we have an example of an operational qualification, or OQ data table. This table summarizes the key measurements and results during the OQ phase, which is critical for verifying that the system operates as intended under normal conditions. The main point here is that clear acceptance criteria, like the temperature tolerance we mentioned, together with well-organized OQ data, help us demonstrate that the equipment is functioning properly. This is essential for compliance and for ensuring product quality. Connecting this to our overall theme, having defined criteria and detailed data supports a robust validation process, giving confidence that our processes are reliable and reproducible. Now, let's talk about performance qualification, or PQ. The main goal here is to demonstrate that the DHS can reliably sterilize or under actual load conditions. Essentially, we need to confirm that the sterilization process works effectively when it's applied to real-world loads. During PQ activities, we perform several critical tests. First, we look at loaded chamber heat distribution and heat penetration studies. These help us understand how evenly heat is distributed throughout the chamber and whether it reaches all products adequately. Next, we conduct biological indicator or endotoxin challenge studies. These are vital because they verify that the sterilization process can effectively eliminate or neutralize microbial contamination or endotoxins. We also test worst-case loading patterns. This means we deliberately load the chamber in ways that are most challenging for heat transfer, ensuring the process remains effective even under less than ideal conditions. Now, why does PQ matter so much? It's because it ensures that the way we load and configure products won't compromise the heat transfer or the sterilization lethality. This step is crucial for maintaining product safety and efficacy. It ties back to our overall goal, guaranteeing that our sterilization process is both reliable and consistent, protecting patient safety and meeting regulatory standards. Let's talk about the biological and endotoxin challenges involved in dry heat sterilization. First, for biological indicators, we typically use organisms like Bacillus atrophius with at least 1 million spores. These are placed in the cold spots of the sterilizer to ensure we're effectively demonstrating microbial inactivation throughout the entire process. The goal here is to confirm that the sterilization cycle can reliably kill these hardy spores, especially in the hardest-to-reach areas. Moving on to depyrogenation cycles, the focus is on endotoxins, which are pyrogens that can cause fever if contaminated. In this process, we apply a known amount of endotoxin to the items, then run the cycle. Our objective is to achieve a minimum of a 3-log reduction, meaning we reduce the endotoxin levels by at least 1,000 times. This is critical because it ensures the removal of pyrogens to safe levels, maintaining product safety and compliance. Connecting these points to the overall theme, both biological indicators and endotoxin challenges are essential for validating that dry heat sterilization is effective, not just against microbes, but also against pyrogens. They help us ensure the highest standards of safety in our sterilization processes. Let's take a closer look at the example PQ temperature mapping from our loaded chamber study results. As you can see here, this slide highlights the key findings from our recent testing. The purpose was to understand how temperature distributes throughout the chamber during operation, which is critical for ensuring consistent product quality. What stands out is the variation we observed across different zones in the chamber. These results help us identify any hot spots or cold spots that could impact the stability of the products. By mapping these temperature profiles, we can optimize the chamber design and operation parameters to achieve more uniform conditions. This example ties directly into our overall goal of improving process reliability and ensuring that our systems deliver consistent results. It's a practical demonstration of how detailed temperature mapping provides valuable insights that drive better control strategies. So, understanding these results is essential for maintaining the integrity of our manufacturing processes. Let's talk about the lethality calculation, specifically the FH value, which relates to dry heat lethality. Similar to moist heat, like what we see with F0, dry heat lethality measures the cumulative lethal effect over time at higher temperatures. The core idea here is that FH is calculated as an integral, or a sum, of the lethal effects over the duration of the heat process. Mathematically, it's expressed as FH equals the integral of 10 raised to the power of T minus divided by Z, with respect to time. What does this mean in practice? Well, T is the temperature measured at any given time T, so it can vary throughout the process. Tref is our reference temperature, often in the range of 160 to 170 degrees Celsius, that we compare against. And Z is a crucial factor. It represents the temperature change needed to produce a 10-fold change in microbial resistance, essentially quantifying how sensitive microbes are to temperature shifts. By understanding and calculating FH, we can accurately assess how effective a dry heat sterilization process is over time. This approach helps us ensure safety and efficacy, tying back into the overall goal of our sterilization methods, reliably eliminating microbes through controlled heat exposure. Let's take a closer look at the essential SOPs we need to have in place. First, the DHS operation and maintenance procedures are critical to ensure our equipment runs smoothly and reliably. Next, temperature mapping and calibration is vital for maintaining consistent conditions, which directly impacts product quality. Biological and endotoxin testing and handling are also key to ensuring safety and compliance with regulatory standards. Moving on to routine controls, it's important to regularly calibrate temperature sensors to keep our data accurate. Performing routine monitoring, such as periodic requalification or reviewing temperature charts, helps us verify that everything remains within specified limits. Additionally, implementing change control processes for process or parameter updates ensures that any modifications are carefully managed and documented. Now, let's talk about the essential qualification documents. The IQ report covers installation checks and calibration activities, confirming that equipment is installed correctly and functioning properly. The OQ report involves empty chamber mapping and control tests to validate performance. The PQ report tests the system under actual loading conditions, including heat penetration and biological or endotoxin challenges, to ensure it meets performance specifications. We also need comprehensive SOPs for operation and monitoring, providing clear instructions for routine activities. Calibration certificates and raw data logs are essential for traceability and demonstrating that our equipment remains in calibration over time. Finally, all of this ties back into documentation and GMP compliance. Proper documentation is the backbone of quality assurance, ensuring that every step is recorded and compliant with regulatory standards. This comprehensive approach helps us maintain high standards and ensures product safety and efficacy throughout the process. Let's talk about some common challenges we might encounter during validation. One typical issue is cold spots in the chamber. When that happens, adjusting the airflow or rethinking the load pattern can usually help improve uniformity. Another challenge is unstable temperature control, which often points to the need for equipment maintenance or tuning the controllers to get better stability. And if we experience a failed endotoxin challenge, it's important to review the cycle settings carefully and then restart the revalidation process to ensure everything is compliant. Now, to tackle these challenges effectively, there are some best practices we should follow. First, always use calibrated instruments and validated SOPs. This ensures our data is accurate and reliable. Second, clearly defining the worst-case load conditions helps us validate the system under the most demanding scenarios. Lastly, repeating the PQ cycles 3 times helps us confirm that the process is consistent and repeatable over multiple runs. These steps and practices are key to maintaining the integrity of our validation efforts and ensuring that we meet our quality standards across the board. Let's wrap up with some key takeaways on dry heat sterilizer qualification. First, it's essential to have robust documentation for IQ, OQ, and PQ. These steps ensure that the sterilizer is installed correctly, operates as intended, and consistently achieves sterilization. Next, core tests like temperature mapping, heat penetration, and biological and endotoxin assurances are critical. They help us verify that the sterilization process reaches the necessary conditions throughout the load. Lethality calculations, specifically FH values, are also vital. They support the justification of cycle parameters by quantifying the sterilization effectiveness. Finally, thorough documentation of all these aspects positions your process well for regulatory compliance. It provides the necessary record to demonstrate that your sterilization process is reliable, consistent, and compliant with industry standards. Together, these points highlight how comprehensive validation and documentation underpin a successful sterilization process, ensuring safety and compliance across the board. For further support, please don't hesitate to contact us. We're here to help you with any questions or additional assistance you might need. If anything from today's presentation sparked your interest, or if you'd like more information, reach out to us. We're happy to provide guidance. And with that, I want to thank you all for your time and attention. Your engagement today is truly appreciated, and I hope you found this session valuable. Remember, support is just a message or a call away, so please feel free to connect whenever needed. Thank you again, and have a great day.